build investor audiences for small cap companies, retail and institutional. 33 years, thousands of clients. We are experts in messaging. National TV ad campaigns, investor webinars, digital media, reaching hundreds of thousands of investors. RedChip. We do it well, we do it right, we do it big. RedChip. Call us or email us today.
Recording in progress.
Hello, and thank you for joining today's event with Calidi Biotherapeutics, which trades on the NYSE American under the ticker CLDI. With us today, we have Eric Poma, the Chief Executive Officer, and Andrew Jackson, Chief Financial Officer. We will begin with a brief presentation in a moment, and then we will answer your questions. Users may submit a question at any time by using the Q&A tool at the bottom of the Zoom window. Before we begin, please allow me to read the Safe Harbor Statement. This call may contain forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. All statements pertaining to future financial and/or operating results, along with other statements about the future expectations, beliefs, goals, plans, or prospects expressed by management, constitute forward-looking statements. Any statements that are not historical fact should also be considered forward-looking statements. Of course, forward-looking statements involve risks and uncertainties. I now turn the webinar over to the Calidi team. Eric, please go ahead.
Thank you so much, and let me start by thanking RedChip for hosting us, and let me start by thanking everyone who dialed in today. I want to take this opportunity to talk about Calidi Biotherapeutics. We think we have an extraordinarily exciting opportunity here with our new RedTail platform, and I just wanted to spend some time to go through that. We really think this is the future of genetic medicine. It's been brought about by a decade of genetic engineering here that I'll take you through. We think this is apropos for cancer, and we think there's the potential to expand this into other indications as well. So I'll walk through the slides here and then be happy to take questions at the end. Our science at Calidi is always evolving. We started in this space about a decade ago working on oncolytic viruses. If you pay attention to the field, you know right now that oncolytic viruses have shown some really nice activity in oncology settings. The problem has always been, though, that you have to directly inject them into the tumor. If you try to deliver them systemically through the bloodstream, they're eliminated immediately by the immune system reacting to the foreign particles that the oncolytic viruses represent. So you can deliver them directly into a subset of patients that have tumors that are directly injectable. But again, that's a subset of patients. The majority of oncology patients with metastatic disease have lesions that are not accessible this way. Calidi years ago started pioneering the use of taking oncolytic viruses and loading them into stem cells. All of our data has shown that that can really enhance the efficacy over traditional oncolytic virus approaches like what you see with CG Oncology or Candel. We have a couple of drugs that are in the clinic or going into the clinic. Neuronova is in a trial at City of Hope for relapse refractory glioblastoma, where we're seeing some really promising data. And we just opened an IND with our compound Supernova for the treatment of advanced solid tumors. Again, these are viruses loaded into stem cells. They do a really nice job of inoculating the virus and inculcating the virus and protecting against immune clearance. But the problem we've seen in all of our models is that the size of these compounds, the stem cell with the virus loaded into it, is too large to really spread efficiently through the body. We're seeing these stem cells get hung up in vasculature in the lung and not really migrating out to the tumors. That's why we've been doing intratumoral delivery with Neuronova and Supernova. But it did raise this really interesting question to us, which was the human cell membrane here really protects the virus from being cleared. Could we just take the virus and instead of putting it into a cell, just wrap it with the cell membrane from a cell that would block immune clearance? The reason for that is that this virus then enveloped with the cell membrane would be about 10,000 times smaller than a stem cell. And that was the genesis of the thinking around RedTail. And I'll take you through that in a little bit more detail here because I think this evolution has been really fascinating. So again, we've been working for over a decade on vaccinia virus. We think this is the ideal virus for delivering genetic medicines and for doing gene altering here. We've created a form of vaccinia virus that is highly selective to only replicating in tumor cells and lysing tumor cells. So if you could do systemic administration, we know that this virus would only really replicate in tumor cells. Because vaccinia virus is a large virus, there's a large capacity for imputing into it DNA encoding other genetic medicines. Because vaccinia virus only replicates in the cytosol, there's not a concern like what you see with lentiviruses where you may get genomic integration. So we've further evolved the vaccinia virus we work with to be able to express multiple different genetic payloads, and I'll show you the data on our lead compound in a minute, but we think this is a really nice step forward. But where we really start to get into uncharted territory here, where we really think we're driving something extraordinary, is we've been able to select for a form of vaccinia virus that retains the host cell membrane of whatever cell line you culture it in. And so here we've done a lot of work determining which are the best cell types to grow the vaccinia virus in so that when it takes on that cell membrane, it's well hidden from the immune system. So this was a huge insight for us. This is a lot of our IP. We've gone one step further where we've engineered the virus to express CD55 along with this cell membrane derived from the host cell. If you know immunology, CD55 is the protein that normal cells express to really avoid immune targeting. So from steps 1 to step 4, we'll be able to create a virus that is really only replicative in tumor cells, only specific for tumor cells. Once inside tumor cells, it can potently lyse them. We've built in the potential to carry genetic medicines that would only be expressed in those tumor cells because their expression is dependent on viral replication. We've packaged that virus now in a human cell membrane that's been optimized for avoiding immune clearance. Now you have an extraordinarily small virus, 10,000 times smaller than stem cells, that is protected from immune clearance. We've gone one step forward to express CD55 on the cell surface to further block any potential for complement-mediated clearance. And that's kind of culminated in CLD401. This is a compound that we've developed that we're moving towards the clinic. Systemic administration in all of our animal models for the first time. We're seeing very, very good dissemination into metastatic sites. I'll show you those data in a minute. And here, another thing we're really excited about is when it replicates in metastatic tumor sites, it expresses extraordinarily high levels of IL-15 superagonist. You don't see that IL-15 superagonist expressed outside of the tumor. There's no detectable IL-15 superagonist in serum, so we're really excited about what we're seeing here in terms of in situ delivery of IL-15 along with the oncolytic virus. This is our first compound coming out of the Redtail platform. We're moving it forward as aggressively as we can. We hope to have an IND filed by the end of next year and move into the clinic. Again, we're still not done here. We continue to evolve the platform here. We've been playing around with multiple payloads being delivered by one molecule, different payloads here. We've also been playing around with getting other proteins expressed on the cell membrane here via the virus. That would be more targeting as well so that we can look at other tumor types and potentially even go outside of oncology. So huge amount of excitement. This platform took 10 years to build, but now we're getting to the point where we'll be moving it into the clinic in the near term here. We're really excited about CLD401. And again, just to show you a little bit more of the data, a little bit more of why we're so excited, CLD401 has the potential for, instead of being limited only to tumors where you can do intratumoral injection, here we can deliver the virus systemically, just through a vein injection here, IV. The virus goes everywhere, goes to metastatic sites, only replicates in the metastatic sites. It's protected from immune clearance by the cell membrane that expresses CD55. Once in the tumor cells, it lyses the tumor cells. That's an immunologic cell death, so you get this immune priming event. But on top of that, we're expressing really high levels of IL-15 superagonist. And I'll go through the superagonist data in a minute here, but what that really does is take that initial priming event from the tumor lysis by the virus and ratchet it up by signaling for activation of T cells and NK cells and driving a memory response to tumor. So here we think we have something wholly unique, systemic delivery, homing to tumor metastatic sites, replication and lysis within the tumor sites to prime the immune system, and then high levels of IL-15 superagonist being expressed in that primed environment to drive memory T cell response, NK activation. Here's just some data. Again, we started working on the non-envelope form. We hit upon creating this envelope form, and then we went one step further to doctor that envelope form to express CD55. We're seeing that the virus that expresses CD55 in the envelope form is highly, highly resistant to immune clearance by human complement. In animal models where we have metastatic disease, we see that the virus alone, this is before we have it express IL-15 superagonist, but the virus alone can find metastatic sites in the mouse and clear them really efficiently off of a systemic administration. So again, this idea that it's able to survive in the serum, it's not cleared immunologically, and it's able to home to metastatic sites in the mouse and clear those sites. This is another model where we had multiple different tumor types in the same mouse, again, representing multiple different metastatic sites. You see very, very nice clearance of those lesions after just one injection. So this was the base virus that we're using here, but again, we wanted to go one step beyond that and deliver genetic medicine here as well. I think one of the most exciting approvals to come recently has been the approval of IL-15 superagonist. So we're understanding that IL-15 superagonist can really alter the tumor microenvironment and drive CD8 and NK cell activation. If you look at the data from Immunity Bio here, they were using IL-15 superagonist in BCG non-responsive, non-muscle invasive bladder cancer. And the reason we think the data is so strong there is because they're delivering that locally. So this is an indication where you can intravesically inject the bladder with extraordinarily high levels of the superagonist. They are also giving back BCG, so that's the priming event. Even though these patients had already progressed on BCG, they're giving back BCG with the IL-15 superagonist to, again, prime the immune system to take off when IL-15 superagonist is presented. So we think this is very akin to what we're doing. When Anktiva had gone into non-small cell lung to give it systemically, though, they had to drop the dose 80-fold because you cannot give this drug you cannot give IL-15 superagonist systemically without running into a lot of toxicity problems. They're shielded from that in the bladder setting. When they go systemic, they have to really drop the dose. When we look at that, we think there's a way to kind of square the circle here by only delivering IL-15 superagonist in high concentration to metastatic tumor sites. So this is where we're really excited about the potential for genetic medicine with this scaffold. Here's just an example in our mouse models. Here you have mice which have two different tumors implanted, left flank, right flank. We're giving CLD401 here, and you can see that there's extraordinarily high levels of IL-15 superagonist expressed in the tumors, but you're really not seeing anything expressed in the serum. So again, this is that potential to only deliver high levels of IL-15 superagonist to metastatic sites without exposing the entire patient to IL-15 superagonist. So this is, again, extraordinarily exciting to us. And when you look at what's actually happening in these mouse models, here I'm showing you untreated animals where the tumor takes off, animals that only received the red tail virus, and you have a slowing of tumor growth. You have one animal cured of the tumor. But when you put in 401, which is the red tail virus that expresses the IL-15 superagonist, you see 80% of the mice cured. So again, because we're expressing such high levels of IL-15 superagonist in the tumor, you're seeing that that effect in mice is able to clear tumor because you're inducing that memory T cell response here along with a viral lytic effect. So very, very exciting about data here. We're excited to bring this compound into the clinic. And again, we think what we've hit on here is the red tail virus, our proprietary genetically modified form of vaccinia virus here. We're excited because it's non-integrating. There's not the risk of transformation like you see with lentiviruses. It has a large insertion capacity, so we can put in multiple genetic medicines here. We understand how to genetically engineer this virus. For our first compound, we're putting in IL-15, moving that to really alter the tumor microenvironment along with the initial immunological priming event of the virus lysis of tumor cells. We're seeing in the mouse models how that really drives an immune effect and really enhances efficacy. Again, we're excited about this compound going into the clinic. There's a lot more to be done here in terms of the platform as we kind of advance it. add additional payloads, make the cell membrane more programmable to allow for enhanced targeting. And all this is being done on a scaffold here where cost of goods is not like cell therapy in the hundreds of thousands to millions of dollars. Cost of goods here is very similar to what you would have for just a standard antibody or protein therapeutic. So we think all this advance, all this kind of innovation here is coming on a very feasible cost of goods that looks really good from a market standpoint. Again, lead compound here, CLD401. We're moving through IND enabling studies, hope to have the IND open by the end of next year. This is, again, the red tail virus. It's encapsulated with cell membrane. It expresses CD55, and when it gets to the tumor and begins to lyse tumor cells, it expresses really high levels of IL-15 superagonist. We think this is an extraordinarily unique compound. We think that going into the clinic here for indications like relapse refractory non-small cell lung, triple negative breast cancer, kidney, melanoma is really appropriate for this kind of approach here. We expect to see data early in the phase 1 study, validating our approach here. We're hard at work at the company in terms of developing additional versions of the technology here, precision oncology, membrane targeting here, additional payloads, and we think the potential to even move outside of oncology into diseases like autoimmune. Pipeline here is really exciting. Neuronova's in the clinic right now. Supernova is moving into the clinic. These are older versions of the technology, still requiring intratumoral injection, but we've seen that intratumoral injection for a subset of patients can be extraordinarily beneficial, and we think we have a superior approach here. Redtail is moving forward rapidly. That's the company's focus right now. CLD401, the IL-15 superagonist expressing form should be in the clinic, hopefully by the end of next year. And we're hard at work on discovery phase for a couple of other compounds here that take advantage of the programmable membrane, that take advantage of additional payloads, and that start to look outside of oncology. Meanwhile, at the company, we continue to try and make sure we're using capital efficiently and being strategic here. Companies had a refresh where we brought in an entirely new management team. I joined the company a few months ago based on the promise of Redtail. Travis Clifton, Chief Medical Officer, joined shortly before I did. Now we have a new chairman of the board here. All of this kind of put in place to drive value. Our team here has done a really nice job reducing debt. We've gone from greater than $8 million on our balance sheet to essentially being debt-free by the end of the year or early next year. We've been reducing our operating costs. Really, G&A expenses have been reduced by 34%, and we continue to look for ways to reduce G&A so that we can focus all of our capital on drug development here. We're advancing the pipeline forward as rapidly as we can. And again, we think that this is a highly innovative technology that will engender discussions with pharmaceuticals around potential to use the Redtail platform in collaboration. So I'll stop with that and happy to take any questions.
Thank you very much, Eric. Press the Q&A button in the at the bottom of your Zoom window and type in your question. Or you can email us at cldi@redchip.com. As we can take only your written questions today, we ask that you please not use the raise hand button. Thank you very much. First question, Eric. Can you elaborate on what makes Calidi's Redtail platform uniquely effective compared to other viral vector platforms, especially in terms of immune evasion and systemic delivery?
Yeah, I think there's two major points I would make. The first is the part around immune evasion. Because of the membrane encapsulation and the CD55 expression, what you have is the potential to deliver virus systemically and have it hone to sites, distal sites of disease. We've seen that viruses can have a great effect. Oncolytic viruses can have a great effect when delivered intratumorally, but that's a subset of patients. Here you have the potential to kind of go into the vast majority of patients that have metastatic disease that's not injectable. You have that potential with Redtail. And then the other thing that we think is extraordinarily exciting is you have the potential to, when you are at those distal metastatic sites, induce the expression of an additional genetic payload that can further enhance the effects, the lytic effects and the immune priming effects of the virus. So the first compound here, IL-15 superagonist, we think is an ideal cytokine to be presenting at high degrees in tumor metastatic sites. So we think that 1-2 combination of being able to get to the metastatic sites, have the oncolytic lytic effect, the immune priming effect, and then express genetic medicine, we think is leaps and bounds ahead of what's currently out there for oncolytic viruses.
What milestones can investors expect as you progress CLD 401 toward phase 1?
Yeah, I think you'll see us put out additional data at major medical meetings on the compound and on the data we're seeing. We're hard at work at getting GMP manufacturing up and running. It's something the company has a lot of experience with. We've done this multiple times. Most recently, we got an IND open on Supernova. So it's blocking and tackling right now. We'll release additional data at medical meetings showing the utility of CLD401 in different animal models. But also we're, again, very, very hard at work on getting GMP manufacturing done. If anybody out there has followed an IND, you know that the CMC section is usually the hardest. We've got a lot of experience here. That's where our focus is right now.
Regarding potential future applications in autoimmune diseases, could you expand on how the Redtail platform can play a role beyond oncology?
Yeah, so what I'm showing you in these data with 401 is that we can design a virus that specifically replicates only in tumor cells and can deliver a payload. We believe it's possible to engineer a virus that replicates in activated B cells, for instance, where we know that B cell depletion as a strategy for autoimmune is extraordinarily interesting and has driven a lot of clinical benefit. But we think we can also go again one step further, and as you're depleting B cells, you may be able to express an immunosuppressive cytokine. Obviously, in tumor cells, you want an immunoactivating cytokine, but in an autoimmune disease, can you express an immunosuppressive cytokine that tamps down locally inflammation? So we think that, again, the basic tenets of the platform are you can find distal sites of disease through systemic administration, and once there, you can express genetic medicines. We think that will hold true, or that could hold true for autoimmune as well.
Could you discuss the scalability of the Redtail platform, especially concerning commercial manufacturing cost efficiency compared to traditional biologic therapies?
Yeah, so again, I think that's one of the things that really attracted me to the Redtail platform and Calidi is that we're not talking about cell therapy cost of goods here. We're talking about manufacturing costs that look very similar to what development of an antibody in phase 1 or development of a recombinant protein would look like. So there's not the bottleneck in terms of COGS that you usually associate with cell therapy here. So that I think means that this is eminently scalable, right? This is a process and a procedure that we think will be very comfortable to pharma and very scalable here.
Turning now to some financial questions, Eric. Can you give an explanation for the recent warrant inducement offering in simple terms?
Yeah, we were seeing a lot of volume in the stock, a lot of liquidity in the stock, a lot of interest in the stock, and we had some inbound requests from shareholders if they could reprice the warrants or exchange the warrants for lower priced warrants. We thought that was a good way of minimizing dilution, but putting some cash on the balance sheet for us. So we went ahead. It's nice to see that investors have repeat interest in the company. So we were just opportunistic there to add to the balance sheet a little bit in a way that we didn't think was particularly dilutive.
Eric, you've significantly reduced debt and general and administrative expenses. How does this impact your runway?
Yeah, look, I mean, I think we want to make sure we're husbanding our capital here. So we're always looking for ways to reduce costs. And as you reduce costs, you expand runway. And it allows us to just put our resources towards drug development. Again, we think CLD401 is groundbreaking technology. We're quite confident here that we have access to capital when we need it. It's our fiduciary responsibility to show investors that we're spending that capital really in as cost efficient a manner as possible. So yeah, we will continue to look to reduce costs. We think we have good runway at the company, and more importantly, we think we have good access to capital at the company.
Final question, Eric. Could you give us the value proposition? Why should investors take an interest in Calidi right now?
Yeah, look, I think Calidi is doing something very, very unique here. We've developed a platform for the systemic delivery of this proprietary virus here. It can home to tumor sites. It can express genetic medicine. We think you're going to prove out a lot of the scaffold's clinical benefit very early in clinical development. Again, very early in phase 1, if you see activity in a metastatic non-small cell lung cancer patient, we think it goes a long way to proving out the utility of the drug, the utility of the scaffold. So we think we're rapidly moving to a huge inflection point for the stock here. And again, as we put more data out, the data I showed you literally came from the American Society of Clinical Oncology last month. So this is new data. As we put it out, we're starting to attract more interest from investors. We're starting to see more interest from pharma companies. So we think that there's a really nice story here where as the platform has matured, now you're starting to see catch up among investors and among the industry in general as we put out more data.
Thank you for that, Eric. Participants, for more information about Calidi Biotherapeutics, reach us at 1-800-RedChip or email us at cldi@redchip.com. Please visit RedChip's investor information page for Calidi. It's cldiinfo.com. There you can view and download the investor presentation and fact sheet and sign up for news alerts on Calidi. Thank you again to our many participants today. And thank you very much, Eric.
Thank you so much. Thanks, everyone.